right, so let's go over chapter 8, cellular respiration. And again, this will be another chapter where I find you a resourceful YouTube video to go through this cycle with you. All right. And again, I'll continue to tell you when you need to put a star by something. So, cellular respiration is a process that breaks down nutrient molecules produced by photosynthesizers with the component production of ATP. It consumes oxygen and produces CO2. So, cellular respiration, you can remember our respirations, we breathe in oxygen, put out CO2. So cellular respiration does the same. They consume oxygen and put out CO2. It is an aerobic process, which means it uses oxygen. If it were anaerobic, that would mean it did not use oxygen, but it's an aerobic process because it uses oxygen. So it usually involves the complete breakdown of glucose to CO2 in water. So again, you see we're still working with the same things that we worked with photosynthesis, our CO2 and our H2O. So energy is extracted from the glucose molecule, because remember, photosynthesis puts out a glucose molecule, okay? So remember that picture I showed you in chapter 6, where we take glucose and water and put it together. And that gives us, we go through cellular respiration because it's the opposite of photosynthesis. So oxidation reduction enzymes include NAD plus and FAD as coenzymes. So again, we're still working with those redox reactions. It's a picture. Okay. So now we're going to introduce some more terms. We've already talked about, G3P, BPG, all that. So we're going to add in some more. So there's NAD plus. So nicotinamide adenine dinucleotide. That's what NAD plus stands for. As a coenzyme of oxidation reduction, it oxidizes when it gives up electrons, reduces when it accepts. So each NAD plus molecule is used over and over and over again. So then we have FAD, which is flavin adenine dinucleotide. So that's also a coenzyme of your redox reactions, and sometimes through this process we'll use FAD instead of NAD+. So this accepts 2 electrons and 2 hydrogen ions to become FADH2. All right. So cellular respiration has phases, and there are 4 of them. We have glycolysis, the preparatory reaction, the citric acid cycle, and the electron transport chain. So we're going to touch on each one of these, but then I'm going to find you a video that puts it all together and shows you the cycle. So to start with, we have glycolysis, which is the breakdown of glucose into 2 molecules of pyruvate. So you can remember glycolysis is the breakdown of something because lysis, lysis, means to break something down, to lyse it, to cut it in half. So glycolysis turns glucose into something called pyruvate. This happens in the cytoplasm. So this is when ATP is formed, and it does not utilize oxygen. So it's considered anaerobic because it doesn't need oxygen. Remember, overall cellular respiration is aerobic because it has to have oxygen, but the step of glycolysis, you do not need it. So the next step, or our prep reaction, both molecules of pyruvate are oxidized. So both molecules of pyruvate lose some electrons and enter to the mitochondria. So electron energy is stored in NADH. 2 carbons are released as CO2, 1 from each pyruvate. And this slide has a star beside it, as does this one. So then the next step, we're talking about the citric acid cycle. This occurs in the matrix of the mitochondria, and this produces NADH and FADH2. So remember, FADH is the combination of FAD and NAD+. So the citric acid cycle is a series of reactions that ultimately release 4 carbons as CO2. So it turns twice per glucose molecule. There's 1 glucose per each pyruvate. So each turn, you get 1 pyruvate from that. So you also get 2 ATP molecules per glucose. So you put in 1 glucose, you get a pyruvate, and you get 2 ATP. So then you have the next part, so your electron transport chain, which is a series of carriers on the cristae of the mitochondria. So this is where the extraction of energy from NADH and FADH2 happens. It passes electrons from high to low energy states, so it follows that electron gradient chain, high to low. So produces 32 or 34 molecules of ATP by the end of it. Okay, let's see what we got next. All right, so this one has a star beside it as well. So now we're going into glycolysis by itself. So we're going to focus on glycolysis first before we go into any of the other steps. So again, glycolysis happens in the cytoplasm, not in the mitochondria. This is outside of it. There's a series of 10 total reactions, and each one has its own enzyme. So remember, those enzymes, they catalyze these reactions, they keep them going, they push them forward. So there's an energy investment step where you have 2 ATP, so 2 energies are used to activate this glucose. And then the glucose splits into 2 G3P molecules. So you should recognize this G3P because with photosynthesis, we just talked about that's our 3-carbon molecule that comes from the splitting of the 6-carbon molecule that are Rubisco joined our 5-carbon RUBP and our CO2. So I just basically said all of that backwards to work back to see where you get these G3P molecules from. So that's the energy investment step, and then the energy harvesting step. So then that's where the oxidation or the electron loss of G3P happens. So we remove those electrons and those hydrogen ions. 2 electrons and 1 hydrogen ion are accepted by NAD+, resulting in 2 NADH. So 4 ATP are produced by something called substrate level ATP synthesis. So this ATP synthesizes and gives you 4 of them. So there's a net gain of 2 ATP because there are 4 ATP produced minus 2 ATP consumed. And then both G3P molecules convert to something called pyruvate. So that was a mouthful. This will make more sense when you watch it in a cycle. So this is a breakdown of what are the ins and the outs of glycolysis. So we start with our 6-carbon glucose here. You know, we end up with 2 pyruvates. We start with our 2 NAD+. We end up with 2 NADH. We have our 2 ATP. We end up with our 2 ADP. And then, you know, at the end we have a net gain of 2 ATP. So there's a star beside this because this lays out perfectly what goes in and what comes out. So let's see, I skip a bunch of slides. And this, if you would like to read this explains glycolysis actually pretty well. So there's your 6-carbon molecule right there that you get from your RUBP and your CO2 coming together with Rubisco. So there's the beginning of that. And then there's your ATP turning into ADP, adding that phosphate. You know, and it further breaks down into this 6-carbon chain, turns into 2 3s. You know, further breaks down into this, and it just keeps going and going and going. So, all right, we skipped that. All right, so fermentation. This has a star beside it as well. So that's how you make wine. Wine sits in a barrel and ferments for years and years and years. Just an example. So fermentation is an anaerobic process that reduces pyruvate to either something called lactate or alcohol and CO2. So NADH transfers its electrons to this pyruvate, and then alcoholic fermentation carried out by yeasts produces CO2 and ethyl alcohol. So the ethyl alcohol is what gets you drunk. So there you go, using the production of alcoholic spirits and breads. So when you know, back in the day, people would make their own bread. Some people still do. It's all a fermentation process. So lactic acid fermentation carried out by certain bacteria and fungi produces lactic acid or lactate. So that's where you get your cheeses and your yogurt and your sauerkraut and stuff from. And so other bacteria produce chemicals anaerobically, including isopropanol, butyric acid, propionic acid, and acetic acid. So that's a process of fermentation. You don't need to know that. So here's some advantages and disadvantages of this fermentation process. So, an advantage, it gives a quick burst of ATP energy from your muscular activity, right? So when muscles are working vigorously for a short period of time, lactic acid fermentation provides ATP. So that is why lactic acid buildup is not good for your body or your heart. Some disadvantages, lactate and alcohol are toxic to your cells. We already know you drink too much alcohol, you can, give yourself alcohol poisoning and die, things like that. Your lactate can change your pH and cause muscles to fatigue. So there we go, we're going in that lactic acid, so it puts you in an oxygen debt, so you don't have enough oxygen to breathe. So that's why if you have high lactic acid levels, they're not good. Your heart might not have enough oxygen in it to pump and breathe and do its thing. We'll skip the bottom part, but yeasts die from the alcohol they produce by fermentation. There's a star beside this in case I forgot to put that out. Here are the ins and outs of fermentation, just in case you read that other slide where it laid it out. So then we move on to the next phase. We talked about glycolysis, and now we're moving into the preparatory reaction, or the prep reaction. So this is what connects glycolysis to the next step, to the citric acid cycle. So each product of glycolysis, pyruvate, enters to the mitochondria. So pyruvate is a product of glycolysis. So pyruvate is then converted to a 2-carbon acetyl group. So this 2-carbon acetyl group attaches to something called coenzyme A. And when it's that 2-carbon acetyl group joins coenzyme A, they are then formed acetyl-CoA. So you're just putting the names together. So electrons are then picked up by NAD+ and produce NADH. And our CO2 is released and transported out of our mitochondria back into our cytoplasm. This happens this occurs twice per glucose molecule. So remember, just like glycolysis, how many glucoses you put in determines, what you're getting out. And there's a star beside this one as well, and there's a star beside this one. So then, after we've gone through glycolysis, the prep reaction, and now we're into the citric acid cycle. This is also called the Krebs cycle. I'll write that down so I can make sure to look that one up for you. So Krebs cycle, or citric acid cycle. This happens in the mitochondria. Begins with the addition of a C2 acetyl group from the acetyl-CoA to a C4 molecule. Oxalacetate? Not sure. We're going to call it C4 molecule. And this forms a C6 molecule. So it forms citric acid. NADH and FADH2 capture energy electrons. ATP formed by the substrate level phosphorylation. It turns twice for one glucose molecule, one for each pyruvate. And then ultimately, the citric acid cycle produces 4 CO2, 2 ATP, 6 NADH, and 2 FADH2 per glucose molecule. So that's kind of the takeaway of the slide, is that last bullet point of what the citric acid cycle puts out. And so this is the citric acid cycle, but like I said, I'm going to find you a video that explains it much better. And there's the ins and outs of that. All right, so let's talk about 3 more terms, and that'll be it for this chapter. So metabolism, we touched on this in chapter 6. So foods are a source of energy-rich molecules, carbs, fats, proteins. That's all in your diet. They're all good for your metabolism. So then there are degradative reactions, which is catabolism, which break down things They tend to be exergonic. They tend to release energy. Remember, we said in chapter 6 that an exergonic reaction, the energy is exiting. So that happens when you have a degradation or a degradative reaction, catabolism. So then we have synthetic reactions, which is anabolism, which build our molecules, and they tend to be endergonic. They consume that energy because the energy is entering. They're entering. Okay. So deeper into catabolism, glucose is broken down in cellular respiration through glycolysis, prep, citric acid cycle, things like that. Fat breaks down into glycerol and 3 fatty acids. Amino acids break down into carbon chains and amino acids. Deamination, which is NH2 is removed, happens in the liver. Your liver makes ura. I'm trying to see if I want y'all to know this or not. So know these top 3 lines, that in catabolism, glucose is broken down. In cellular respiration, fat breaks down into glycerol and 3 fatty acids, and amino acids break down into carbon chains. Because there's not a star by this slide, and there's not a star by this slide either, or this one. So we'll skip these 2, no catabolism and no metabolism, and then we'll call it on that. All right, so next up is chapter 9.